Somewhere beneath the last traces of sunlight, animals are living in conditions that should make almost every familiar body plan impossible. The pressure is immense, the darkness is complete, and yet one fish is hovering there with a head so transparent you can see directly through its skull. Inside that clear dome, two green eyes glow in the darkness. They are not positioned where you expect eyes to be. They sit inside the skull, pointing upward, watching for shapes above, and they can rotate. Scientists first described this fish in 1939. For decades, they thought they understood its anatomy. They were wrong. Every specimen they examined had been brought up dead, and something remarkable about the animal had been destroyed before scientists could see it. Then there is a shark whose jaw does not stay where you expect it to. When it attacks, the jaw shoots forward from the skull, catches its prey, and snaps back into place in a fraction of a second. The entire movement takes about 322 milliseconds. This shark has been built this way for 125 million years. And then there is an eel with a mouth so enormous that it can look larger than the rest of its body. It hunts where there is almost no light, at depths where the pressure would crush an unprotected vessel. Another animal has evolved a way to hunt using a color of light its prey cannot see. And one of the ocean's most famous ugly creatures has another problem entirely. The animal in the photograph millions of people know is not what it actually looks like. It is a corpse. The strange appearance was created by the physics of dragging a deep sea animal from roughly 1,200 meters, which is about 4,000 feet, toward the surface. Down where these animals actually live, the bodies make sense. The transparent head, the rotating eyes, the launching jaw, the enormous mouth, the strange light, none of them are random. None of them are broken. They are solutions to a world where sunlight disappears, pressure becomes extreme, and finding food can mean surviving with almost no information at all. And that is where the deeper mystery begins. Because the more closely scientists examine these animals, the less they look like mistakes. They begin to look like the logical result of living somewhere almost nothing else can survive. Today, we're going deeper into these strange animals to understand how bodies that look so completely wrong can make perfect sense in the world they evolved to survive. There is a point in the ocean where the surface world stops arriving. Sunlight weakens as it travels downward, its colors disappearing one by one, until at roughly 200 meters, about 656 feet, the remaining daylight has become too faint to support the familiar rhythms of the upper ocean. By 1,000 meters, about 3,280 feet, sunlight has effectively vanished. Below that boundary, the deep sea begins to take on its own physical rules. The first of those rules is pressure. At the bottom of the Mariana Trench, nearly 11,000 meters, about 36,000 feet beneath the surface, the surrounding water presses with a force of roughly 109 megapascals, about 15,750 pounds per square inch. At sea level, atmospheric pressure is about 101 kilopascals, roughly 14.7 pounds per square inch. The difference is enormous, but the animals living there do not experience it as a sudden weight. Their bodies have developed within that pressure, with tissues and fluids arranged so that the water outside and the water inside remain in balance. Then there is the absence of sunlight. It is not simply a darker version of daytime. There are no dawns or sunsets at these depths, no seasonal return of illumination, and no hidden beam reaching the seafloor from above. Natural sunlight becomes irrelevant long before the deepest trenches begin. What remains is a world where vision, when it exists, must operate on the faintest traces of available light, or on flashes produced by living organisms themselves. Food presents another constraint. The surface ocean is constantly creating organic material through sunlight driven life, but only a small portion of that material survives the descent. Dead plankton, fragments of tissue, fecal particles, and other organic remains drift downward in a slow snowfall known as marine snow. Along the way, bacteria and other organisms consume much of it. By the time the particles reach great depths, what remains is sparse enough that an animal may spend enormous stretches of time waiting for the next useful meal. The deep ocean is therefore divided into broad ecological regions according to changing light, pressure, temperature, and available food. The twilight zone extends from roughly 200 meters, about 656 feet, to 1,000 meters, about 3,280 feet. Beneath it lies the midnight zone, reaching down to approximately 4,000 meters, about 13,100 feet. Farther below are the abyssal depths, extending to around 6,000 meters, about 19,700 feet, followed by the Hadal depths of the deepest trenches. These boundaries are not walls. They are gradual changes, layered across an enormous volume of water that covers most of the planet. Temperatures generally fall, pressure rises, and meals become increasingly uncertain as depth increases. Yet life continues through every layer, with bodies shaped around conditions that would be fatal to creatures from the surface. For the animals living here, pressure, darkness, cold, and scarcity are not temporary obstacles. They are the background of existence, present through every generation. Their bodies have been refined within these limits for millions of years, until features that seem extraordinary from above become ordinary below. Even the shape of a body, the way it floats, and the amount of energy it spends staying alive can become solutions written directly into the anatomy of an animal. Somewhere in those depths, one of the first creatures to challenge our assumptions about what a deep sea body should look like is drifting through the water, carrying a reputation it never deserved. That creature is the blobfish. For a creature that became famous around the world, the blobfish has spent remarkably little of its life looking like the animal people recognize. Its familiar face belongs to a moment of violence, not to its existence in the deep ocean, where its body has a completely different purpose and form. In 2013, a public poll organized by the Ugly Animal Preservation Society gave the blobfish an unlikely distinction, naming it the world's ugliest animal. The photograph circulated everywhere, showing a pale body, sagging features, and an expression that seemed almost human. Yet the photograph captured a body removed from the conditions that had shaped it. The animal had been brought upward from roughly 1,200 meters, about 3,900 feet, where the surrounding pressure was vastly greater than at the surface. This change is known as barotrauma. As pressure falls during a rapid ascent, gases dissolved within tissues can expand, while structures adapted to intense compression lose their normal form. For the blobfish, the journey from its habitat to the surface can transform the proportions and texture of its body so completely that the result becomes almost unrecognizable. The famous photograph therefore tells us more about the journey upward than about the fish itself. Below the surface, Psychrolutes marcetus has a different presence. Its body is soft and comparatively delicate, with little dense muscle and a skeleton that provides far less rigidity than that of many familiar fishes. Rather than carrying a heavily reinforced body through the water, it makes use of the surrounding pressure and the buoyancy of its own tissues. The flesh contains a high proportion of water and other low density material, leaving the animal only slightly denser than seawater. That arrangement matters because the blobfish does not possess the gas-filled swim bladder used by many shallow water fishes to regulate buoyancy. At extreme depth, a gas-filled chamber would be a liability. Instead, the blobfish can remain close to the seafloor without constantly swimming to support itself. Its lifestyle is built around conserving energy in a place where food is sparse and movement can be costly. The name blobfish is also shared by more than one species. Cycrolutes marcitus occurs around southern Australia, Tasmania, and New Zealand, while Cycrolutes microporos is found in nearby deep waters. Their lives unfold far below the surface, beyond the range of ordinary observation, where their unusual softness becomes useful rather than defective. Their metabolism is thought to be extremely slow, matching an existence in which opportunities for feeding can be separated by long intervals. Some estimates have suggested lifespans reaching more than a century, although the precise age of wild blobfish remains difficult to establish. Even the pace of its life is connected to the same economy that shapes its body. Energy is not spent on speed, rigid structure, or constant movement when the environment rewards endurance instead. The photograph that made the blobfish famous is therefore a strange kind of biological illusion. It shows a real animal, but only after the physical conditions that gave its body meaning have been removed. Its sagging features are not the appearance evolution produced for the deep. They are the aftermath of bringing a deep sea body into a world with radically different pressure. Far below the surface, the living fish remains adapted to its own surroundings, suspended close to the seabed in a form that makes sense there. The creature that became a symbol of ugliness was never really seen in the photograph that made it famous. What was captured was a body after its world had been taken away. For decades, the barreleye existed in scientific literature as a fish that seemed to have a perfectly ordinary face. The mistake was not in the fish, it was in what happened to the fish before anyone could truly see it. Macropinna microstoma was formally described in 1939 based on specimens collected from deep water in the North Pacific. Those preserved bodies showed a small, dark fish with an unusual head, but nothing suggested the extraordinary structure that living animals carry. The most important part had disappeared during collection. A transparent, fluid-filled dome covers the upper portion of the barreleye's head, extending over the delicate eyes like a clear biological shield. Under the pressure of its natural habitat, that dome retains its shape. During ascent, the tissues deform and collapse, leaving behind a specimen that can resemble an entirely different kind of fish. For generations, researchers also misunderstood the animal's eyes. Two dark structures positioned above its small mouth appeared to be eyes, and early anatomical descriptions treated them that way. They are actually olfactory organs used for detecting chemical traces in the surrounding water. The real eyes sit much deeper inside the transparent dome, long cylindrical structures filled with a greenish pigment. Their resting position is particularly unusual. They point upward, toward the faint illumination filtering down from the world above. In the barreleye's habitat, that direction contains one of the few useful visual backgrounds available. A small creature passing overhead can interrupt the remaining glow and become visible as a dark shape. The barreleye can therefore spend much of its time looking into the water above while remaining nearly motionless. Then, living observation changed the anatomy completely. In 2009, researchers from the Monterey Bay Aquarium Research Institute observed barreleyes using a remotely operated vehicle. The cameras revealed something that preserved specimens had never shown. The eyes were not permanently fixed in their upward position. They could rotate forward, allowing the fish to shift its gaze toward prey as it moved into position. What had appeared to be a strange arrangement of immovable organs became an adjustable visual system. The green coloration of those eyes also has a useful optical function. A yellow pigment in the lenses filters incoming wavelengths, helping the fish distinguish faint natural illumination from the flashes and glimmers produced by bioluminescent animals. In the dim water, that filtering may help reveal prey whose own light would otherwise blend into the surrounding darkness. The transparent dome provides another advantage. Barreleyes are associated with gelatinous siphonophore colonies whose tentacles carry stinging cells. The dome may protect the sensitive eyes from those stinging structures, while the fish searches around the colony for small prey. Its most remarkable feature is therefore not simply transparency. The same structure can provide protection while leaving the eyes free to collect scarce light. The barreleye remains a small animal, reaching about 15 centimeters, about 6 inches in length. It has been recorded at depths from roughly 610 meters, about 2,000 feet, to nearly 790 meters, about 2,600 feet, where its unusual anatomy fits the conditions around it with remarkable precision. For 70 years, the scientific record contained a distorted version of this fish. The living barreleye revealed that the missing feature was not an oddity added to its design, but the part that made the entire design understandable. Its transparent head, rotating eyes, and strange upward gaze belong together, forming one visual system shaped around a world where even the faintest trace of light can become useful for the animal below. The goblin shark does not need to look fast to hunt fast. Its body drifts with an almost delicate appearance, while the long, flattened rostrum extends ahead like a sensory field. Along this snout are ampullae of Lorenzini, tiny electroreceptive pores capable of detecting the faint electrical signals produced by contracting muscles. A buried crustacean, a fish moving through the water, or another animal hidden nearby can leave an electrical trace, even when there is no useful visual image to follow. That makes the snout less like a spear and more like an instrument for locating opportunity. The shark can approach without the rapid pursuit associated with many predators. Much of its body is soft compared with the muscular forms of faster sharks, and its swimming is not built around long chases. Energy is precious in deep water, so the body has little reason to spend it unnecessarily. Then the face changes. The goblin shark's lower jaw can move far beyond the normal outline of its head, unfolding from its resting position through a remarkable sequence of skeletal and ligament movements. High-speed recordings revealed that the jaw is propelled outward by elastic structures working with the surrounding musculature. Rather than simply opening its mouth, the shark projects the entire biting apparatus toward its prey. The recorded maximum speed of the jaw was about 3 meters per second, about 10 feet per second. During a strike, the upper and lower jaws can travel forward together, extending as much as roughly 1/10 of the shark's body length. The movement is among the most extreme examples of jaw protrusion known in sharks, transforming a seemingly passive animal into a predator capable of reaching well beyond its head. A complete strike unfolds in less than half a second. The jaws extend, close around the target, and return toward the skull in a sequence too brief for the eye to follow naturally. The mechanism is powered by a combination of muscles, flexible tissues, and ligaments that store and release mechanical energy. What looks bizarre from the outside is therefore a carefully coordinated system of leverage. The advantage becomes clearer when the rest of the animal is considered. A goblin shark does not need to accelerate its entire body toward prey. Its head can remain relatively close to the target while the jaws perform the final movement. Reach substitutes for pursuit, allowing the shark to capture food without paying the energetic cost of a sustained chase. Its appearance carries another small deception. The skin has a distinctive pinkish coloration, but this is not produced by a conventional layer of bright pigment. Blood vessels beneath the translucent skin contribute strongly to the color, giving the shark an almost otherworldly appearance when viewed outside its natural surroundings. Mitsukarina austoni belongs to a lineage with roots stretching back roughly 125 million years. Fossils and living relatives show how ancient this branch of shark evolution is, although the idea that the species itself has remained completely unchanged is too simple. Its anatomy has persisted because its particular combination of traits continues to function within deep water habitats. Adults can reach about 3.8 meters, about 12 and 1/2 feet, although many individuals encountered by researchers are smaller. Records have come largely from deep water fisheries, especially around Japan, meaning much of what science knows about this shark has still been learned from animals encountered after capture. Yet its most remarkable feature is visible only when the shark feeds. A face that seems almost fragile becomes a launching mechanism, extending the mouth far beyond the skull before returning it to place. The goblin shark does not need the speed of a hunter that chases. It has developed another way to close the distance, storing its advantage in the architecture of its own face. A gulper eel can make its own body seem like an afterthought. Most of its proportions are devoted to a long, slender tail and a small frame until the mouth opens and the entire shape changes. The lower jaw stretches outward, the thin tissues of the throat unfold, and the opening becomes so broad that the animal appears briefly to be little more than a mouth suspended in water. Europharynx pelicanoides is also known as the pelican eel, a name inspired by the loose, pouch-like throat that resembles the expandable bill of a pelican. It lives across tropical and temperate oceans, generally between about 500 meters, about 1,640 feet, and 3,000 meters, about 9,840 feet. At those depths, a large jaw does not need to be strong in the way a predator's jaws are strong near the surface. The gulper eel is not built for crushing. Its jaws contain little muscle, and the surrounding tissues are remarkably delicate. Instead, the mouth works more like a drifting collection surface. When opened broadly, it can increase the area through which water passes, allowing small crustaceans, fish, and squid to be gathered when they happen to move within reach. A powerful bite would provide little advantage when prey is often small and scattered. What matters is the ability to accept whatever arrives. That same economy appears throughout the animal. Its skull is surprisingly small compared with the enormous oral cavity. The body lacks the sturdy appearance of many familiar fishes, with no scales and relatively little muscle beneath its dark, velvety skin. Small eyes sit toward the front of the head, suited to an environment where vision has limited information to work with. Near the opposite end of the animal, another structure adds an entirely different dimension. The gulper eel carries a bioluminescent organ near the tip of its long tail. The glowing tissue can act as a lure, creating a small point of living light in the surrounding water. The eel can move its tail while holding its mouth open, bringing potential prey toward the expanding cavity rather than spending energy pursuing them. Its stomach follows the same principle of flexibility. After feeding, the digestive tract can expand considerably, allowing the eel to accommodate a meal that would seem impossibly large compared with its narrow resting body. In a habitat where opportunities to feed can be separated by long intervals, The ability to make use of an unusually large meal may be more valuable than maintaining a body designed for constant hunting. The animal itself can reach about 1 meter, about 3 feet, in length, although much of that measurement belongs to the tail. Its proportions make more sense when the body is viewed as a system of separate tasks. The tail provides movement and carries the luminous organ. The narrow torso minimizes unnecessary mass. The mouth waits for opportunity. In 2018, an expedition aboard exploration vessel Nautilus encountered a juvenile gulper eel within the Papahanaumokuakea Marine National Monument. Cameras captured the animal expanding its enormous throat in the water, revealing how dramatically the tissues could unfold around its pharynx. Unlike preserved specimens, the living animal showed the movement itself, turning an anatomical curiosity into a functioning behavior. The gulper eel's strange proportions are therefore not a collection of unrelated oddities. The tiny skull, enormous mouth, expandable stomach, slender body, and glowing tail all belong to one strategy built around a simple condition of deep water. When food arrives unpredictably, the animal that can gather more of an unexpected opportunity may not need to chase anything at all. Light in the deep ocean does not come from above. It rises from within the water itself, appearing as brief sparks, soft glows, and small points that drift through the darkness. Across much of the deep sea, these signals are so common that bioluminescence has become one of the dominant forms of communication and survival. Scientists estimate that roughly 75% of deep sea animals are capable of producing their own light. The exact proportion varies depending on how researchers define the deep sea and which groups are included, but the prevalence is extraordinary. In a habitat where sunlight disappears, evolution has repeatedly arrived at the same solution, making living organisms into sources of illumination. The chemistry behind that glow is remarkably efficient. Many animals produce light through a reaction involving luciferin, a light-producing molecule, and luciferase, an enzyme that helps the reaction occur. The result is known as cold light because very little energy is released as heat. Some creatures manufacture the necessary chemicals themselves. Others maintain partnerships with bioluminescent bacteria housed inside specialized organs, allowing the bacteria to provide a continuous source of illumination. A single flash can serve very different purposes. Some animals use light to draw prey within reach. Others produce sudden bursts that startle a predator or interrupt its attack. Mating signals can help individuals locate one another across open water, while carefully arranged patterns may allow members of the same species to recognize each other. Some animals even use light as a form of illumination, briefly revealing nearby objects that would otherwise remain hidden. The color itself is part of the strategy. Blue wavelengths travel farther through seawater than most other visible colors, which is why blue dominates deep-sea bioluminescence. Many animals have visual systems tuned to that narrow range, making them especially sensitive to the flashes produced by their neighbors. In this way, light and vision have evolved together, each shaping the usefulness of the other. The scale can become difficult to imagine when individual flashes combine into larger events. Along the ocean surface, enormous concentrations of bioluminescent bacteria can sometimes create what sailors have described as milky seas. The water appears to glow across vast areas, sometimes covering thousands of square kilometers, roughly thousands of square miles, with a diffuse blue-white radiance. These events can persist long enough to be observed by satellites, turning microscopic organisms into a phenomenon visible from far above the planet. For much of scientific history, however, This living light was largely absent from the record. Specimens brought upward in nets could preserve chemical traces of bioluminescent organs, but they could not show a flash, a lure, a defensive display, or the way an animal adjusted its light in response to another creature. The behavior belonged to the living ocean, and it disappeared before researchers could watch it. That changed as remotely operated vehicles began carrying cameras into deeper water. During expeditions in the late 20th century, researchers started seeing organisms produce light in their natural surroundings, revealing that bioluminescence was not a collection of isolated curiosities. It had evolved independently across more than 40 major lineages of marine life, appearing again and again as different animals encountered similar problems. Each lineage arrived at its own version of the same language. A brief pulse could mean opportunity. A glow could conceal a body. A flash could signal a mate, confuse a predator, or reveal prey. In the deep ocean, where ordinary landmarks are scarce, light became more than something to see. It became information moving through the water, carrying messages from one living body to another across an otherwise open world. From beneath, a hatchetfish can almost disappear without moving. Its body is compressed from side to side, giving it the thin profile that inspired its name, but its most important feature lies along the underside. A row of tiny light-producing organs turns the fish into something closer to a moving patch of the surrounding water than a solid object. The habitat of the hatchetfish occupies the twilight zone, where enough sunlight remains to create a faint glow above the animal. That remaining illumination creates a peculiar geometry. A predator looking upward sees the brighter water behind a potential meal. Any opaque body crossing that background forms a dark outline, and even a small silhouette can reveal exactly where prey is swimming. The hatchetfish answers this problem with counterillumination. Its photophores, specialized organs containing bioluminescent tissue, shine downward from the underside of its body. The emitted light can be adjusted in intensity, allowing the fish to approximate the brightness of the water above it. Viewed from below, the body no longer produces the same dark interruption against the background. The outline becomes difficult to separate from the surrounding glow. This is not simply a matter of turning a light on. The animal must constantly respond to changes in the faint illumination reaching its depth. Clouds, surface movement, changes in depth, and the position of the animal itself can alter the amount of light coming from above. Its visual system and light-producing organs therefore work together, adjusting the signal as conditions change. For a long time, researchers imagined that the fish achieved this effect by reflecting incoming light from its body like a small mirror. That explanation was appealing because it seemed mechanically simple. But biological light is more complicated. Studies involving researchers at the University of Pennsylvania showed that hatchetfish skin can scatter light diffusely rather than returning it in one precise direction. That scattering creates another advantage. A predator may use its own bioluminescent light as a search beam, sending a flash across the water to reveal nearby prey. Instead of producing a bright reflection that exposes the fish, the hatchetfish can redirect some of that incoming illumination through its tissues and toward its underside, where the photophores help disperse it downward. The animal effectively turns an attacker's searchlight into part of its own camouflage. The arrangement also changes with depth. In the dim region where sunlight still provides a background, counterillumination is useful because there is something against which a silhouette can be measured. Farther down, that background disappears, the strategy becomes less valuable, and the hatchetfish's large, upward facing eyes take on another role. They become sensitive detectors for the faint, bioluminescent signals of animals above. This creates a delicate division of attention. During one part of its vertical range, the fish is concerned with preventing predators below from seeing it. In another, it uses the same upward orientation to search for food. The eyes that help conceal the body can also locate the next meal, while the organs beneath the body regulate how visible that body appears to anything watching from below. Most hatchetfish remain small, generally only a few centimeters, about a few inches, long, yet their survival depends on controlling something far larger than their bodies. The contrast between themselves and the water around them. Their camouflage is not a coating placed over the animal. It is an active process, produced moment by living tissue. In a world where visibility can determine whether a predator finds its next meal, the hatchetfish does not need to become faster or stronger. It changes the light itself until the space where its body should be contains almost nothing that gives it away. Red light disappears quickly in seawater, and that simple physical fact has shaped an entire hidden advantage for one deep-sea predator. As sunlight travels downward, its longer red wavelengths are absorbed more readily than blue and green light. Far below the surface, red becomes almost absent from the natural visual environment. For most animals living there, this means red illumination offers little information. Their eyes are tuned to the wavelengths that remain available, while red falls outside the useful range. The loose-jawed dragonfish, Malacosteus niger, occupies this perceptual gap with remarkable precision. Beneath each eye are specialized photophores capable of producing red bioluminescence. Instead of broadcasting the familiar blue glow used by many other deep-sea animals, the dragonfish projects a longer wavelength that most potential prey cannot detect. The surrounding water can therefore be illuminated without creating an obvious warning signal. To the dragonfish, the effect is like carrying a private source of illumination. Its own eyes contain adaptations that allow them to respond to red wavelengths, so the animal can use its red light to examine nearby water while much of the surrounding community remains unable to perceive the beam. A small fish that might react instantly to a visible blue flash can continue moving normally while the dragonfish studies its outline. The advantage depends on an unusual partnership between chemistry and vision. The light-producing organs are positioned beneath the eyes, placing the illumination close to the animal's visual field. Red-sensitive photoreceptors allow the dragonfish to receive information from wavelengths that are largely unavailable to other deep-sea predators. The result is not merely a different color of bioluminescence. It is a private, visual channel. Even more unusual chemistry appears inside the eyes. The pigments associated with red sensitivity are related to bacteriochlorophyll, a family of light-absorbing molecules associated with certain photosynthetic bacteria. In an animal that lives far from sunlight, such chemistry has become part of a sensory system instead of a photosynthetic one. The evolutionary history behind this adaptation is complex, but its function is clear enough. A molecule associated with capturing light has been repurposed to help a predator detect light. Its red glow also changes the meaning of distance. Blue bioluminescence can announce an animal's presence to many observers, but red light travels only a limited distance through seawater. That keeps the dragonfish's unusual signal relatively local, useful for examining the immediate space around its head without turning the entire neighborhood into a beacon for other predators. Some species within the dragonfish group carry another remarkable feature in the same small head. Their teeth can become so long and slender that they extend beyond the mouth when the jaws are closed. Rather than forming broad, crushing surfaces, the teeth interlock like a delicate cage, helping secure prey once it enters the mouth. The dragonfish itself is not large, and its body is built for a world where conserving energy matters. Its hunting system does not require a bright display or a rapid pursuit. Instead, it uses a wavelength that has become scarce in the environment, then equips its own eyes to see what that wavelength reveals. There is something elegant about the asymmetry. The ocean has removed red from the ordinary visual landscape, and the dragonfish has turned that absence into an advantage. Its prey does not need to be blind. It only needs to lack the particular sensitivity that the predator possesses. In that narrow difference between what one animal can see and another cannot, an entire hunting strategy becomes possible. The dragonfish does not overpower the visual world around it. It simply occupies a part of that world that almost everything else has left unused. The first thing to notice about a female deep-sea anglerfish is not the lure, but the stillness of the hunting strategy built around it. She does not need to search across a wide area or pursue passing animals. Her body is arranged around a single point of invitation, a small light suspended above a mouth designed to wait. Female serratioid anglerfishes belong to a diverse group containing roughly 160 known species. Their forms vary considerably, but the basic arrangement remains familiar. The head is large compared with the rest of the body. The mouth opens broadly, and slender teeth point inward from the jaws. These teeth do not function like grinding surfaces. They act more like retaining hooks, allowing prey that has crossed the threshold to remain inside. Above the mouth rises a modified dorsal spine called the elysium. At its end sits the esca, a specialized structure that produces the lure's glow. In many species, bioluminescent bacteria live within the esca and generate the light. The fish can influence the appearance of the signal, making the lure seem like a small living object moving through the surrounding water. That detail matters because the light is not identical across anglerfish species. Some escas resemble tiny lanterns. Others appear more like glowing beads, delicate filaments, or small worm-like forms. Natural selection has shaped these structures around different prey communities, so the lure becomes a specialized visual invitation rather than a generic beacon. A creature drifting nearby may approach because the light resembles something worth investigating. The anglerfish itself remains almost motionless while this happens. Its energy expenditure can stay low because the lure performs much of the searching. Instead of moving through the water looking for food, the fish creates a small moving signal and waits for curiosity to bring another animal within reach. The distinction is subtle, but biologically important. Hunting becomes a problem of attraction rather than pursuit. Once prey reaches the mouth, the anatomy changes the odds further. The jaws can open widely, while the teeth angle toward the throat. A struggling animal may find that each movement carries it deeper rather than toward an exit. The mouth and throat are also capable of expanding, allowing some anglerfish to consume prey remarkably large relative to their own bodies. The stomach shares this flexibility. Its walls can stretch as food enters, temporarily accommodating a meal that would seem disproportionate to the fish's slender body. In an environment where feeding opportunities are uncertain, the ability to accept a large meal when one appears can provide a valuable reserve of energy. Female anglerfish remain small by ordinary surface standards. Some species reach around 20 centimeters, about 8 inches, while others are smaller. Their compact size, expandable tissues, inward-pointing teeth, and luminous lure form a single feeding system, each feature supporting the same low-energy method of survival. There is also a deeper peculiarity in the lure itself. Because its light comes from living bacteria rather than sunlight, the ESCA represents a partnership between two organisms. The anglerfish provides a protected home and access to nutrients, while the bacteria supply the illumination that helps attract prey. What appears to be one glowing organ is therefore a small ecosystem contained within the body of a predator. The female anglerfish has no need for speed when attraction can replace pursuit. She can remain suspended while the lure moves, the jaws wait, and the stomach stands ready for whatever arrives. Her strange appearance is therefore not a collection of unrelated abnormalities. The enormous mouth, specialized teeth, expandable body, and living light form one remarkably economical way of feeding in a place where every unnecessary movement carries a cost. For the male serratioid anglerfish, adulthood is less a destination than a transformation already written into his body. He emerges from the egg as a tiny, free-swimming animal, carrying almost none of the machinery needed for a life of independent feeding. His mouth, digestive system, and musculature remain greatly reduced, while his sense of smell becomes unusually important. The contrast with the female is striking. Some males measure only 1 centimeter, about half an inch, while females can be many times larger. Their proportions are shaped less for roaming than for finding the one partner on which their adult existence depends. Instead of developing a luminous lure, the male develops enlarged olfactory organs, allowing him to detect chemical traces released into the surrounding water. In a habitat where individuals may be separated by vast distances, scent becomes a thread connecting one small body to another. When the male finally encounters a female of the same species, the encounter changes the course of both lives. He attaches by biting her skin, and in many serratioid species, specialized tissues begin a process of permanent fusion. The boundary between the two animals gradually becomes less distinct as their tissues connect and their circulatory systems join. This is not merely a long attachment, it is a biological merger. After fusion, the male no longer needs to maintain the full anatomy required for independent existence. His eyes and other structures can degenerate, while his body becomes increasingly specialized for producing sperm. What began as a separate fish becomes a living reproductive organ carried by the female. Several males can remain attached to one female, creating an arrangement unlike the mating systems familiar from most vertebrate life. The female gains a dependable source of sperm without having to find another mate each time reproduction becomes possible. For the male, attachment transforms a dangerous search into permanent access to a reproductive partner. The deeper biological puzzle lies within the immune system. In most vertebrates, cells from another individual are recognized as foreign because their surface markers do not fully match. This recognition is essential for defending the body against pathogens, yet it also creates the central obstacle of organ transplantation. Tissue from an unrelated donor can trigger an immune response powerful enough to destroy the transplanted organ. Serratioid anglerfishes have evolved an unusual solution. Their genomes show major changes in components associated with adaptive immunity, including reductions in some mechanisms used by other vertebrates to recognize and attack foreign cells. The result is an immune system capable of tolerating a naturally occurring fusion between genetically different individuals. Research published in 2020 by scientists from the Max Planck Institute of Immunobiology and Epigenetics and the University of Washington helped reveal how unusual this adaptation is. Instead of simply weakening immunity everywhere, these fishes appear to have reorganized parts of a defense system that, in other vertebrates, depends on recognizing genetic individuality. That discovery gives the tiny male a significance beyond the darkness where he lives. His body demonstrates that vertebrate immunity is not a single fixed design, but a system evolution can reshape under unusual pressures. A creature measuring only 1 centimeter, about half an inch, can therefore preserve a biological solution to a problem that medicine still wrestles with. The male's transformation is difficult to separate from the environment that produced it. Finding a mate is so uncertain that natural selection favored a strategy in which discovery could become permanence. Once contact occurs, separation is no longer part of the story. Two bodies become one functioning reproductive system, with the male surrendering nearly everything that once made him an independent animal. The strawberry squid carries its two eyes as though they belong to different worlds. One is large and rounded, the other noticeably smaller, and their imbalance is not an accident of growth. It is a response to a life lived between two directions of danger, where light from above and light from below carry entirely different information. The larger eye points upward, toward the faint illumination descending through the water column. Its great size gives it a larger opening for gathering scarce photons, helping the squid detect the brief flashes produced by animals moving overhead. In this part of the ocean, those flashes can reveal both potential meals and nearby predators. The eye is therefore shaped around sensitivity, gathering whatever traces of blue light reach it from the upper water. Below the squid, another kind of signal is present. Many animals use counterillumination to soften the dark outline they would otherwise cast against the faint glow overhead. Their own blue light can make them difficult to distinguish from the background when viewed from beneath. The strawberry squid has an unusual response to this problem. Its smaller eye points downward, and a yellow filter in that eye changes the light entering the visual system, increasing the contrast of the blue bioluminescence against the surrounding darkness. The result is an arrangement that resembles two separate optical instruments sharing one nervous system. One watches the sky of the deep sea, the other surveys the space beneath the animal. Neither eye needs to perform every visual task equally well, because evolution has divided the work according to direction. That division becomes especially useful in the mesopelagic zone, where the squid spends much of its life. This region receives enough sunlight to create a faint background glow, yet not enough to reveal the shapes of most animals clearly. Bioluminescence fills the remaining visual landscape with brief signals, scattered points, and moving traces. A creature passing overhead and another glowing beneath the squid can present completely different optical problems within the same moment. The squid's body adds another layer of adaptation. Its red coloration is difficult for many deep water animals to distinguish because red wavelengths are absorbed rapidly by seawater. At depth, red tissue can therefore appear very dark, helping the squid blend into the surrounding water. Across its body are numerous small photophores, the light-producing organs that give the animal its distinctive strawberry-like appearance. Its mantle can reach about 13 centimeters, about 5 inches, making the animal modest in size, despite the elaborate visual system carried within its head. The species is believed to be widespread and abundant in the twilight zone, although much of its natural behavior remains difficult to observe directly. Much of what is known comes from specimens and occasional encounters with living animals brought into view by deep ocean exploration. What makes the strawberry squid especially interesting is not simply that its eyes are different sizes. Their asymmetry records two separate ways of seeing the same habitat. One eye searches the faint upper field for movement and flashes. The other specializes in the lower field, where prey may attempt to disappear using the same luminous language that dominates the surrounding water. Even the unequal proportions make practical sense when viewed this way. The large eye is not simply an oversized version of the smaller one, and the smaller eye is not a damaged copy of its companion. Their differences are functional, reflecting the different kinds of light that arrive from opposite sides of the animal. In the deep ocean, anatomy can become a record of direction, with form shaped by where information comes from. The animal does not need one perfect eye. It needs two specialized answers to two different directions, carried together on a single body. At first glance, the giant siphonophore seems to have been drawn as a single drifting creature, a long translucent body suspended in the midwater. Praia dubia can extend to about 46 meters, about 150 feet, placing it among the longest animals known to science. Yet its length belongs to no single body. It belongs to a living arrangement built from hundreds of genetically identical units, each carrying out one narrow task. These units are called zooids. Some propel the colony by contracting specialized structures that push water backward. Others capture and digest food. Still, others produce offspring, while defensive units protect the colony with stinging cells. Along the trailing portions, specialized structures can form an elaborate feeding network, turning the surrounding water into a broad field of potential contact. No single zooid contains the complete machinery of an independent animal. A propulsion unit cannot simply detach and become a swimming creature. A feeding unit cannot wander away and hunt on its own. Their survival depends on remaining integrated with the rest of the colony, where thousands of small biological actions become one coordinated way of living. The resemblance to an ordinary animal becomes stronger when the colony begins to move. Propulsive zooids work together, creating a coordinated rhythm that carries the entire structure through the water. Feeding structures respond to drifting prey, digestive units process what has been captured, and reproductive units maintain the colony's future. There is no central brain directing each movement, yet the colony behaves with a coherence that feels remarkably familiar. Biologists have long wrestled with the language needed to describe such a body. Calling it a colony emphasizes that it is assembled from distinct clonal units. Calling it a superorganism emphasizes that those units have become so interdependent that the whole behaves as a single living system. Neither description completely captures the strange middle ground occupied by a siphonophore. The same basic principle appears in the Portuguese man-of-war, often mistaken for a jellyfish. It is also a siphonophore, although its colony is organized differently from its deep-sea relatives. What looks like one floating animal is instead a collection of specialized zooids, whose combined functions create something larger than any member could accomplish alone. Deep in the water column, some siphonophores can gather into expansive feeding formations. Their tentacles may extend across broad spaces, creating delicate curtains of stinging structures that intercept small organisms drifting through the current. The arrangement turns the colony itself into a moving net, with different zooids contributing to the same biological process. Its form can remain flexible, bending with currents, while specialized units continue their separate work without losing coordination. That network also creates unusual relationships with other animals. Barreleye fishes are believed to approach siphonophore colonies and take advantage of prey caught among their tentacles. Their transparent head structures may offer protection from the stinging cells, allowing them to remain close to a colony while feeding on what the siphonophore has already gathered. The deeper significance of the siphonophore lies in the way individuality becomes difficult to define. Each zooid begins from the same genetic blueprint, yet specialization separates their roles so completely that the colony can resemble an organ system assembled from hundreds of animals. Movement, feeding, defense, and reproduction no longer belong neatly to separate individuals. They belong to the living structure created by their cooperation. Seen this way, the great length of Praia dubia is not simply a measurement of one animal's body. It is the physical expression of many bodies functioning together, each surrendering independence in exchange for a role within something much larger. The first impression comes from the armor. Bathynomus giganteus carries a body divided into overlapping plates, each segment forming part of a design that could have been lifted from the floor of a garden and enlarged beyond recognition. The resemblance to a common pillbug is not accidental. Both belong to the order Isopoda, sharing the same basic body architecture despite living in radically different worlds. A fully grown giant isopod can reach about 50 centimeters, about 20 inches, from end to end. Its smaller terrestrial relatives may measure only a few centimeters, about an inch, yet the essential arrangement remains familiar. 14 walking legs move beneath the armored body, while compound eyes gather the sparse visual information available around the seafloor. The rigid outer covering provides protection, but it also creates a physical limit. To grow, the animal must periodically shed that shell and form a larger one, a process that becomes increasingly demanding as its body expands. The species was first described scientifically in 1879, after specimens from the deep Atlantic revealed an animal unlike the smaller isopods already familiar to naturalists. Since then, Bathynomus giganteus has been recorded across parts of the Atlantic, Pacific, and Indian Oceans, generally along the ocean floor, from roughly 150 meters, about 500 feet, to more than 2,100 meters, about 7,000 feet. Down there, the isopod lives by accepting uncertainty as part of its biology. It does not need a steady supply of food. Instead, it is adapted to the irregular arrival of material sinking from above. A dead fish, a large piece of organic matter, or the remains of a much larger animal can become an unexpected banquet on an otherwise sparse stretch of seafloor. When such a meal appears, the isopod can consume far more than it could use immediately. Much of that surplus becomes stored energy within lipid-rich tissues. Its body becomes a reservoir, holding the remains of a brief period of abundance through whatever interval follows. That interval can be remarkably long. One giant isopod maintained in captivity in Japan survived for more than 5 years without eating. During prolonged fasting, its metabolism can fall dramatically, reducing the energy required to maintain its body. The animal does not need to roam continuously in search of another meal. Its physiology allows time itself to become part of the strategy. There is something especially old-fashioned about this way of surviving. The fossil history of isopods reaches back more than 300 million years, placing the broader body plan deep in the Paleozoic past, long before the first dinosaurs appeared. The modern giant isopod is not a living fossil in the strict scientific sense, because its lineage has continued to evolve, but its fundamental architecture belongs to a remarkably ancient branch of animal history. Even its defenses have produced unusual encounters. Giant isopods have been observed attaching themselves to capture deep-water sharks, sometimes gripping tissue with enough persistence that releasing them becomes difficult. Their armored bodies and strong limbs are not designed for pursuit. They are built for endurance, opportunism, and making the most of whatever the seafloor provides. Yet size itself remains the most curious part of their story. A familiar body plan has been stretched far beyond the proportions of its garden-dwelling relatives, creating an animal that seems almost oversized for the same basic design. In the deep ocean, where food can arrive unpredictably and temperatures remain low, unusual dimensions can emerge from unusual pressures, and the giant isopod offers one of the clearest examples of that deep sea tendency. Size changes meaning in the deep ocean. A body that would seem oversized near the surface can become an efficient answer to conditions that remain difficult to reproduce anywhere else. This pattern, known as deep sea gigantism, appears across several unrelated branches of marine life, yet its causes are still being pieced together from scattered evidence. The giant squid offers one of the clearest examples. Architeuthis ducks can reach about 13 meters, about 43 feet, in total length, while some shallower relatives remain only around 60 centimeters, about 2 feet, long. Its enormous body is paired with eyes adapted to detect faint disturbances and bioluminescent signals in the surrounding water. Size alone does not explain its existence, but a larger body can provide greater reach, greater reserves, and a larger visual system in an environment where encounters with food may be widely separated. The colossal squid pushes the pattern further in another direction. Messanicoteuthes hamiltoni is generally shorter than the largest giant squid, yet it can be substantially heavier. Its eyes may reach nearly 28 centimeters, about 11 inches, across, giving it the largest known eyes among living animals. Those immense organs gather scarce photons from a habitat where even a small amount of distant biological light can carry useful information. Elsewhere, the Japanese spider crab extends its body plan into an extraordinary span. Its legs can reach roughly 3½ meters, about 12 feet, across, allowing a relatively compact central body to occupy a surprisingly large area. The giant tube worm follows a different route. Riftia pachyptilla, associated with hydrothermal vents, can reach about 2 meters, about 7 feet, while related worms from shallower environments may be measured in fractions of an inch. These animals are not following one shared blueprint. Their lineages arrived at unusual size through separate evolutionary histories. Several explanations have been proposed. One possibility involves oxygen. In cold, deep water, dissolved oxygen can be relatively abundant, potentially allowing some invertebrates to support larger bodies than their relatives could maintain in warmer, oxygen-poor environments. Another possibility concerns predation. Where encounters with predators are less frequent, living longer may become more feasible, giving an animal additional years in which to grow. Cold itself may also favor larger bodies. A larger animal has a lower surface area relative to its volume, which can reduce the rate at which heat is exchanged with the surrounding water. In near-freezing conditions, that relationship may make greater size energetically useful. A large body can also store more reserves, an advantage when the interval between successful meals stretches across long periods. Yet, none of these ideas explains every giant. The giant tube worm depends on a symbiotic relationship with bacteria, while the giant squid hunts actively and the Japanese spider crab moves across the seafloor. Their environments, diets, body structures, and evolutionary histories differ too greatly for a single explanation to fit them all. That is what makes deep-sea gigantism so interesting scientifically. It is not a rule that every animal follows, nor is size simply the inevitable result of living at depth. Instead, the abyss appears to create circumstances in which unusual dimensions can sometimes become useful, allowing certain lineages to cross boundaries that would be difficult to sustain elsewhere. The pattern is widespread, the mechanisms are varied, and the evolutionary story remains open. What matters is that size becomes part of a larger bargain between metabolism, food, temperature, oxygen, and time. In the abyss, growth is not simply an increase in dimensions. It can change how an animal moves, feeds, stores energy, and remains viable across years of uncertain opportunity. In the deepest reaches of the ocean, evolution can leave an organ behind without replacing it with another version of the same thing. The change begins much earlier, during development, when an eye that once carried useful information becomes an unnecessary expense. Photoreceptor cells require energy, neural connections require maintenance, and the tissues surrounding them must be supplied throughout life. When no meaningful light reaches the animal, those costs can become difficult to justify biologically. The process is not deliberate. Natural selection does not decide that an animal should lose its eyes. Instead, once vision stops improving survival or reproduction, mutations that would previously have been harmful may no longer carry much consequence. Across many generations, the machinery of sight can gradually deteriorate. A lens may become malformed, neural pathways may shrink, and genes involved in visual development can disappear from the genome altogether. The deep-sea fish of the genus Ipnops offers an especially unusual example. Its young begin life with functional eyes, yet adulthood brings a different arrangement. The eyes flatten as development continues, their lenses disappear, and the visual apparatus becomes reduced beneath transparent tissue. What looks like a missing organ is therefore the final stage of a developmental transition, shaped by an environment in which ordinary vision offers little advantage. Genomic studies of deep-sea fishes have found similar losses at the molecular level. Hadal snailfishes, among the deepest known fishes, show extensive changes in genes associated with vision, reflecting the long evolutionary history of life under conditions where sunlight cannot provide useful information. Their genomes preserve a record of what the environment has made unnecessary, just as clearly as their bodies preserve what remains useful. Along the sides of many deep-sea fishes, another sensory system becomes far more important. The lateral line consists of rows of pressure-sensitive structures that register disturbances moving through the surrounding water. A nearby animal does not need to be visible for its motion to be detected. Every flick of a fin, every displacement of water, can leave a physical signal traveling through the medium around the fish. Other species rely heavily on chemical information. Enhanced chemoreception allows some deep-sea animals to detect dissolved substances released by prey, potential mates, or other organisms. The information arrives as traces carried by currents, rather than as images formed by light. In some predators, electroreception adds another layer, sensing tiny electrical fields generated by the muscular activity of nearby animals. These senses are not lesser versions of vision. They are better matched to the information actually available. Vision excels when light creates useful patterns. Pressure sensing works when movement disturbs water. Chemical detection works when molecules travel through currents. Electrical sensing works when living tissue generates fields. The environment determines which signals matter, and evolution tends to preserve the machinery that can interpret them. Yet the deep ocean has not abandoned eyes altogether. The barreleye fish retains an elaborate visual system, with tubular eyes that can rotate upward to gather faint light from above and then shift forward when feeding. Its existence reveals an important distinction. Darkness does not make vision universally useless. It makes vision useful only when an animal has evolved a precise way to extract information from the limited light that remains. For some fishes, the most successful adaptation was therefore not a better eye, but the gradual acceptance that an eye was no longer necessary. What disappeared was not perception itself. perception became something broader, distributed through pressure, chemistry, electricity, and movement, allowing an animal to remain deeply connected to its surroundings without relying on the one sense that depends upon light. There is something unusual about an animal whose most important adaptation is not to become a better hunter, but to stop hunting altogether. The vampire squid, Vampiroteuthis infernalis, belongs to a lineage so distinct that it occupies its own order, Vampiromorphida. Its name suggests a predator, yet its way of life is built around gathering what other organisms leave behind. The vampire squid spends much of its life within the oxygen minimum zone, a layer where dissolved oxygen becomes scarce enough to exclude many active animals. In some regions, this habitat lies roughly 600 to 900 meters, about 2,000 to 3,000 feet beneath the surface. For a creature that needs to conserve oxygen, remaining there is less a hardship than an opportunity. Its metabolism is exceptionally low for a cephalopod. Every movement has an energetic cost, and every energetic cost ultimately draws upon oxygen. By reducing activity, the vampire squid can remain within water that would challenge animals with faster metabolisms. Its body is adapted not for pursuit, but for persistence. Food arrives in a completely different form. Tiny fragments of organic material drift downward from productive waters above, carrying pieces of dead organisms, discarded biological material, and fecal particles. Together, these falling fragments form what oceanographers call marine snow. The material descends gradually through the water column, creating a scattered supply of energy far removed from the surface ecosystems that produced it. Instead of chasing individual prey, the vampire squid extends two long, delicate filaments into the surrounding water. These structures can retract into the body when not needed. Particles become attached to the filaments and are transferred toward the arms, where fine cilia move the collected material toward the mouth. Feeding becomes a process of gathering, almost like collecting drifting dust from an enormous moving sky. Its fins provide another economical solution. Rather than relying on rapid jet propulsion, the vampire squid uses broad, rounded structures on its mantle to hover and maneuver through the surrounding water with relatively little expenditure. The animal can change direction without turning its entire body, allowing it to remain suspended while searching for particles carried past its position. Even its defenses reflect the same restrained biology. When threatened, the vampire squid can produce flashes of blue bioluminescence from specialized organs along its body and arms. It can also invert its webbed arms over its body, exposing fleshy projections while transforming its familiar outline into something less recognizable. The display combines light, shape, and movement without requiring a prolonged chase. The result is an animal that has made an unusual evolutionary trade. It does not compete directly with many of the active predators occupying better oxygenated waters. Instead, it inhabits a layer where reduced oxygen limits competition and where falling organic matter provides a diffuse food supply. The very condition that makes the habitat difficult for other animals can therefore create a refuge for a creature built to use little oxygen. Despite its common name, the vampire squid has no blood-drinking habit and no need to attack living prey. Its existence is tied instead to the slow movement of material through the ocean, connecting life near the surface with organisms living far below. In that connection, the animal occupies a narrow ecological space shaped by oxygen, gravity, and the steady descent of organic matter through time. In this way, its survival depends on becoming economical rather than powerful. The vampire squid does not conquer the oxygen minimum zone through speed or force. It persists there by requiring less, moving gently, and gathering what the larger ocean allows to fall. At nearly 11 kilometers, about 7 miles beneath the surface, pressure becomes more than a physical force pressing against an animal. At the deepest parts of the Mariana Trench, around 36,000 feet, pressure reaches roughly 108 megapascals, approximately 15,750 pounds per square inch. For an unprotected human body, the surrounding force would be overwhelming. Inside a living cell, the problem is more delicate because pressure can alter the shapes of the molecules that keep life operating. Proteins are folded chains whose three-dimensional forms determine what they can do. Their surfaces must fit other molecules with extraordinary precision, allowing reactions, transport, signaling, and countless other processes to proceed. Under extreme pressure, those structures can become distorted or collapse into forms that no longer function properly. The danger is therefore molecular rather than merely mechanical. A cell can remain intact while the machinery within it begins to lose its shape. Even enzymes can lose the precise contours needed to bind their chemical partners, turning a microscopic change in form into a failure of an entire reaction. Deep sea organisms have evolved chemistry that helps prevent this. Among the most important compounds are piezolites, small molecules that stabilize biological structures under pressure. One of the best studied is trimethylamine oxide, a naturally occurring compound that interacts with proteins and helps counteract the structural effects of compression. In many deep sea fishes, the amount of this compound increases with habitat depth, creating a chemical adjustment that follows the physical conditions surrounding the animal. This relationship has a remarkable limit. As depth increases, the concentration of trimethylamine oxide required for protein stability also rises. At the greatest depths occupied by fishes, the amount becomes substantial enough to influence the chemistry of their tissues. Scientists have used this relationship to help understand the pressures experienced by deep living species, because their internal chemistry carries a record of the environment outside their bodies. Buoyancy requires another solution. Many fishes near the surface depend on gas-filled swim bladders, which allow them to remain suspended without constant swimming. At extreme depth, however, compressible gas becomes difficult to use. Deep-sea fishes instead rely on tissues containing large amounts of water, reduced mineral density, and lipids that provide buoyancy without creating a fragile pocket of gas. Their bodies become better matched to the surrounding water by containing less material that pressure can compress dramatically. The same principle appears in their skeletons. Some deep-sea fishes have bones and cartilage that are less heavily mineralized than those of their shallow water relatives. In hadal snailfishes, skeletal structures are remarkably delicate, contributing to bodies that appear almost unfinished when compared with familiar fishes from the surface. Yet this softness is part of the solution. A heavily reinforced body would require more energy to build and maintain, while providing little advantage in an environment where the surrounding water supports every part of the animal. Even transparency can belong to this same economy. Reduced pigmentation, watery tissues, and delicate structures can make an animal appear almost insubstantial, but such bodies are not incomplete versions of surface life. They are specialized arrangements shaped by a world where pressure is constant, buoyancy works differently, and ordinary molecular structures need chemical assistance. At the deepest boundaries of the ocean, survival therefore depends on changes too small to see. A molecule protects a protein. A watery tissue replaces a gas-filled organ. A flexible skeleton bends where a rigid one would demand unnecessary strength. The abyss does not simply press against life from the outside. It reaches inward, and evolution answers by changing life at the scale of chemistry itself. A whale can spend a lifetime moving through an ocean that seems almost impossibly large, yet its final destination may become one of the most concentrated centers of life on the seafloor. When the body of a large whale begins its descent, gravity carries thousands of kilograms, about thousands of pounds, of living history into a realm where food is normally scattered across enormous distances. The sinking can take hours or more than a day, depending on the animal and the depth below. The body becomes a meeting place for species that rarely encounter one another. Once the carcass reaches the bottom, the transformation begins with remarkable speed. Sleeper sharks, hagfish, rat-tail fishes, crabs, amphipods, and giant isopods converge on the new source of energy. Their arrival is not coordinated by any central signal. Instead, chemical traces spread through the surrounding water, guiding scavengers toward a concentration of organic material that may have been absent from that part of the seafloor for years. Soft tissue begins disappearing in layers, with different animals feeding from different surfaces and tissues. A large whale can support the removal of tens of kilograms, about tens of pounds, of tissue each day. As the flesh thins, the character of the community changes. Animals that once fed on the exposed body begin giving way to worms and crustaceans that occupy the sediment around the remains. The bones themselves become increasingly important, holding lipids that are inaccessible while the carcass is fresh. What began as a single body gradually becomes a habitat with its own succession of organisms. Inside those bones, another process is unfolding. Bacteria consume stored fats and release hydrogen sulfide as a byproduct. The compound is toxic to many animals, yet certain microorganisms can use it as an energy source through chemosynthesis. Their growth forms the foundation for another food web, one that does not depend directly on sunlight. Bacterial communities spread across the bones, supporting specialized worms, mollusks, and other animals adapted to this unusual chemistry. Some of these creatures resemble organisms associated with hydrothermal vents, where chemical energy also supports dense communities in the absence of sunlight. This resemblance has led scientists to consider a larger possibility. During periods when whales were far more abundant than they are today, their carcasses may have formed temporary biological stepping stones across the sea floor. Organisms adapted to chemically rich habitats could potentially move between distant environments by colonizing one whale fall, reproducing there, and later reaching another. The idea gives a different meaning to migration beneath the ocean. Movement through the deep is not always accomplished by an animal swimming from place to place. Sometimes an ecosystem itself can travel, carried downward in the body of a giant, then rebuilt on the seabed through many generations of organisms. A whale fall may remain biologically active for decades, and the bones can continue supporting specialized communities long after the recognizable animal has disappeared. For a blue whale, the largest animal known to have lived, death does not simply remove biomass from the ocean. It redistributes that biomass into a concentrated habitat, feeding scavengers first, then sediment dwellers, then organisms capable of extracting energy from the chemistry locked inside bone. The sequence can unfold over many years, turning one fallen body into a layered community whose members occupy different stages of the same transformation. In this way, the seafloor receives more than a carcass. It receives structure, nutrients, shelter, and chemical energy gathered into one temporary place. A whale that once carried its own ecosystem through the open ocean can become, after sinking, the foundation for another ecosystem beneath it. At the bottom of the Izu Ogasawara Trench, the shape of a fish becomes almost difficult to recognize. In April 2023, researchers from the University of Western Australia and Tokyo University of Marine Science and Technology sent a camera lander into the trench south of Japan. The instrument descended into water more than 8,000 meters, about 27,000 feet, below the surface, carrying its lenses into a place where very few vertebrates have ever been observed. There, a juvenile snailfish appeared in the frame. Its body was small and translucent, with a rounded head and a soft outline that seemed almost unfinished. Yet this delicate animal represented the deepest confirmed record for a fish and the deepest verified observation of any vertebrate. The depth reached roughly 8,300 meters, about 27,200 feet beneath the sea surface. At that pressure, the familiar architecture of a surface fish would be poorly suited to survival. Snailfishes belonging to the family Liparidae have evolved a body that gives up much of that architecture. Their skeletons are lightly built, their tissues contain abundant water, and their bodies approach neutral buoyancy. Instead of spending substantial energy fighting gravity with muscular swimming, they can remain suspended with relatively little effort. The result is an animal whose apparent fragility is closely connected to the demands of its habitat. Inside the cells, another adaptation becomes even more important. Proteins must retain their precise molecular shapes, while the surrounding pressure reaches levels that can disrupt those structures. Extreme deep-sea snailfishes accumulate unusually high concentrations of trimethylamine oxide, a pressure-stabilizing compound that helps maintain protein function. The chemistry of their tissues is therefore matched to the depth around them, allowing cellular machinery to continue operating where ordinary proteins would become unstable. Their softness also changes the relationship between body and environment. A rigid, heavily mineralized skeleton would require considerable biological investment, while offering little advantage in water that supports the body from every direction. In the deepest trenches, reduced bone mineralization can make the animal lighter, more flexible, and less costly to maintain. The snailfish looks less like a conventional fish because it has surrendered structures that are no longer especially useful. Its food source follows the same pattern of specialization. Amphipods, small crustaceans that inhabit the deepest trenches, can occur in surprising concentrations around areas where organic material accumulates. Snailfishes feed among them, using a flexible body and relatively modest energy demands to exploit a food web that exists beneath the reach of most fishes. Several snailfish species have been recorded deeper than 7,600 meters, about 25,000 feet, showing that the hadal environment is not merely a boundary they occasionally cross. For this family, extreme depth can be a place where populations gather and reproduce, turning one of Earth's most severe environments into a genuine habitat. The record-holding juvenile was not a giant, nor a heavily armored predator. It was smaller than a ruler, translucent and soft-bodied, filmed during a brief encounter with a machine sent into a world humans could not previously observe directly. Its appearance carries a strange lesson in scale. The deepest known vertebrate does not look like an animal built to conquer an extreme environment. It looks like an animal that has become so thoroughly fitted to that environment that almost everything unnecessary has disappeared. What makes the discovery especially valuable is not simply the depth of the record. The fish provides a living example of how far vertebrate biology can be reshaped by environmental pressure. Its form, chemistry, buoyancy, feeding, and growth all belong to the same physical world, each adaptation reinforcing the others. For much of the history of deep sea exploration, the ocean was studied through what could be brought back from it. Nets descended into darkness, swept through the water or across the seafloor, and returned with animals preserved only as bodies. By the time those specimens reached the surface, pressure had fallen, tissues had changed, colors had faded, and postures had been altered. A creature adapted to an extreme environment could arrive on a laboratory table looking like something entirely different. That limitation shaped an entire era of marine biology. Scientists could examine bones, muscles, stomachs, teeth, and organs with extraordinary care, yet the living animal remained outside the frame. Movement could only be inferred. Feeding behavior had to be reconstructed. Defensive displays were invisible. Even the arrangement of soft tissues could be misleading when a body had been compressed, expanded, or distorted during its ascent. The arrival of remotely operated vehicles changed the relationship between observation and reality. These machines carried cameras, lights, and mechanical arms directly into deep water, allowing researchers to watch animals where their bodies were still supported by the surrounding pressure. By the early 1980s, increasingly capable vehicles were opening new possibilities, and later systems reached depths of more than 6,000 meters, about 20,000 feet, while transmitting detailed images to scientists at the surface. Living observations began correcting old assumptions in ways that were sometimes remarkably simple. The barreleye fish had been known from preserved specimens for decades, yet its extraordinary transparent head structure was difficult to interpret without seeing the animal alive. In living individuals, the dome surrounds delicate visual organs and remains an active part of an integrated sensory system. The same shift changed the understanding of the blobfish. Specimens brought upward from great depth could lose their normal shape as pressure fell, producing the distorted appearance that later became famous. Observations of living blobfishes showed a body far better suited to its environment than the familiar surface image suggested. Other animals revealed behaviors that preserved specimens could never provide. The goblin shark, for example, possesses a remarkable jaw that can project forward during feeding, turning an anatomical curiosity into a rapid predatory mechanism. Watching that movement in living animals transformed a strange structure into a functional part of its ecology. The vampire squid offered an even broader correction. Its appearance suggested an active hunter to early observers, yet direct observations showed a different way of living. It drifts through oxygen, poor water, collects falling organic particles, and uses delicate filaments and cilia rather than pursuing prey through the darkness. Its survival strategy depends on conserving energy, not spending it in pursuit. Remotely operated vehicles also revealed that color itself had been misunderstood. A preserved animal may appear uniformly dark after pigments change during collection, while living individuals can display patterns of bioluminescence, reflective tissues, and changing body positions that disappear almost immediately outside their natural surroundings. Behavior became part of anatomy because the way an organism moves can explain structures that seem mysterious when viewed only after death. This has left deep sea biology with an unusual relationship to its own history. Many older specimens remain scientifically valuable, preserving tissues, anatomy, and genetic information that cannot be recovered from brief encounters in the wild. Yet living observation provides something a specimen cannot preserve: context. An animal can be seen moving, feeding, responding to light, interacting with another species, and occupying the exact conditions that shaped its body. Direct observation has therefore transformed the deep ocean from a collection of specimens into a living environment. Even now, only a fraction of that environment has been watched in place, leaving the history of deep sea discovery unfinished, with vast biological communities still known only through traces, fragments, or not at all. There is an odd imbalance in our knowledge of the planet. We have drawn detailed maps of worlds millions of miles away, while broad portions of the seafloor beneath our own oceans remain known only in rough outlines. More than 80% of the ocean floor has not been mapped at a resolution finer than about 5 kilometers, roughly 3 miles. The Moon and Mars have both been charted in greater detail across much of their surfaces. That gap becomes more remarkable when biological surveys begin. In the Clarion-Clipperton Zone, a vast region of the Pacific Ocean extending across an area larger than the continental United States, researchers continue to encounter animals that have never been formally described. A survey published in 2023 found that more than 90% of the specimens collected belonged to species new to science. These were not simply unfamiliar populations of known animals. Many represented organisms that had never existed in a scientific catalog before. The pattern is repeated wherever expeditions spend enough time collecting and observing. New species are common, and sometimes the discoveries reach deeper into the structure of life itself, producing new genera and, on rarer occasions, entirely new families. The reason is not that deep sea biology is somehow separate from evolution. It is that the habitat is enormous, fragmented by depth and geography, and difficult to sample. A single expedition may examine only a tiny patch of sediment, water, or rock, leaving immense surrounding regions untouched. A mapped surface is not the same as an observed ecosystem. A camera may cross a narrow transect and record dozens of forms within minutes, while neighboring sediments remain untouched. A sediment core samples only a small column, and a trawl gathers whatever happens to enter its path. Species living beyond those paths leave no record at all. Even the boundaries of known habitats can shift as instruments reach greater depths and reveal communities occupying places that earlier surveys considered biologically sparse. The history of the giant squid offers a glimpse of what that uncertainty can conceal. For more than a century, its existence was known largely through damaged bodies and fragments recovered from sperm whale stomachs. Then, in 2004, researchers obtained the first confirmed images of a living giant squid in its natural environment. 15 years later, in 2019, another expedition captured the animal in the open, deep ocean, moving through the habitat where it had spent its entire life beyond ordinary human sight. Such discoveries make estimates of undiscovered deep-sea diversity difficult to pin down. Scientists have proposed that hundreds of thousands, perhaps several million, species may remain undescribed. The enormous range between those estimates is itself revealing. There is no dependable inventory of the animals already living below the surface, so calculating what remains becomes an exercise built upon incomplete foundations. The ocean floor stretches across more than 60% of Earth's surface area, forming a continuous environment larger than any continent. Much of it lies beyond the reach of ordinary observation, divided into trenches, plains, ridges, slopes, vents, sediments, and isolated habitats where different communities can persist. Even within places visited by research vehicles, only narrow corridors are seen, and only for brief intervals. What has been found there already includes creatures with unfamiliar anatomy, unusual metabolisms, unexpected behaviors, and evolutionary histories that have changed scientific understanding. Yet, Those discoveries occupy only a thin window into a much larger living world. The remarkable part is not simply that unknown animals remain beneath the sea. It is that every new glimpse suggests the same thing. The deep ocean has never stopped making life that we have not yet learned to recognize. Some of the strangest animals in the deep ocean have become useful not because they resemble anything familiar, but because their biology solves problems that human medicine has struggled with for generations. Their unusual bodies contain experiments written by evolution, showing that systems considered essential can sometimes be altered, simplified, or rebuilt without losing function. The anglerfish offers one of the clearest examples. In species where the male permanently fuses with the female, the tissues of two genetically different animals must remain together without the destructive immune response that would normally attack foreign tissue. Research led by scientists at the Max Planck Institute and the University of Washington found extensive losses in genes associated with adaptive immunity in several anglerfish lineages. The animals appear to have traded part of the immune machinery used to recognize foreign tissue for a reproductive system built around permanent attachment. That unusual compromise has attracted attention from immunologists because organ transplantation faces the same fundamental problem. A recipient's immune system can recognize donated tissue as foreign and attempt to destroy it. The deep sea also offers lessons at a much smaller scale. Proteins must fold into precise shapes to function, yet pressure and other environmental stresses can disturb those shapes. Deep living fish accumulate trimethylamine oxide, a small molecule that helps stabilize proteins and preserve their functional structure. Biochemists study this protective chemistry because protein misfolding is involved in several human diseases, including neurodegenerative disorders. The molecule does not provide a finished treatment, but it offers a natural example of how cells can influence the physical behavior of proteins under difficult conditions. The deeper lesson is that molecular stability is not a fixed property. It can depend on the surrounding chemistry, and changing that chemistry can shift the balance between a useful folded protein and a damaged one. For medicine, that principle is valuable even when the original deep sea molecule remains far from clinical use. Light provides another unexpected connection. The deep sea dragonfish, Malacosteus, Niger, produces red bioluminescence, a wavelength that is poorly detected by many other deep ocean animals. Its retina contains a photosensitizer derived from bacteriochlorophyll, a pigment family better known from photosynthetic microorganisms. Rather than simply copying the visual chemistry of surface animals, the dragonfish has incorporated an unusual light-sensitive molecule into a visual system suited to its own private band of illumination. This biology has helped researchers understand how pigments can be modified and combined to extend the range of light detection, offering ideas relevant to the design of sensitive optical systems. Then there are the microbes living around hydrothermal vents. Some thrive at temperatures and chemical conditions that would damage ordinary enzymes. One of the most consequential discoveries came from Thermus aquaticus, a heat-loving bacterium isolated from a hot spring in Yellowstone National Park. Its heat-stable enzyme, known as Taq polymerase, made repeated cycles of the polymerase chain reaction practical because the enzyme could withstand the high temperatures required during the process. That advance became foundational to modern molecular biology, supporting genetic analysis, forensic identification, medical diagnostics, and countless laboratory techniques. None of these organisms evolved to help us. The anglerfish did not abandon immune functions for transplantation. The dragonfish did not reshape its retina to inspire optical engineering. The vent bacterium did not become heat tolerant for a laboratory machine. Each simply became suited to the conditions around it. Human science arrived later and noticed that these solutions could be borrowed, studied, and translated. In that sense, the deep ocean is more than a catalog of strange creatures. It is a library of biological possibilities, written in molecules, tissues, pigments, and enzymes, with some of its most useful pages belonging to animals that spent their entire lives beyond the reach of sunlight. The deep ocean is sometimes described as a destination, as though life began in sunlight and later wandered downward into a harsher world. Yet for many animals, that story begins in the wrong place. Their ancestors were not visitors learning to endure the abyss. Their lineages were shaped there, generation after generation, until the conditions we might call extreme became ordinary biological surroundings. Consider the blobfish. Its soft, gelatinous tissues can appear strangely inadequate when seen at the surface, where pressure is greatly reduced. Down where it normally lives, that same body is suited to a world in which dense water supports it and rigid structures offer little advantage. The familiar photograph of a collapsed blobfish is therefore less a portrait of the living animal than a record of what happens when a deep water body plan is removed from the pressure that gives it context. Pressure is woven into these animals from the earliest stages of development. Their proteins, membranes, skeletons, muscles, and buoyancy systems are assembled for conditions that would disrupt the physiology of most surface organisms. Bringing such creatures upward is not simply moving them from one habitat to another. It is changing the physical environment in which their cells were built to operate. The goblin shark offers a much older example. Its lineage reaches back roughly 125 million years, carrying a distinctive combination of features through an immense span of geological time. The elongated snout is packed with sensory organs that can detect weak electrical signals from nearby prey. Its protrusible jaws can extend rapidly from the mouth, allowing the shark to seize an animal without relying on speed or a conventional chase. These traits make more sense as parts of a deep water existence than as compromises inherited from a brighter world. Other lineages reveal the same principle in different ways. The giant isopod carries an ancient body plan into modern abyssal habitats, while the vampire squid occupies oxygen-poor water with a metabolism suited to conditions that exclude many other cephalopods. Snailfish have bodies so soft and lightly built that they seem almost unfinished beside familiar fish, yet that softness is closely connected to their ability to function under immense pressure. Their unusual appearance is not evidence of incomplete evolution. It is evidence that evolution has been working toward a different set of physical requirements. Even reproduction reflects this independence. Some deep-sea animals synchronize their lives with scarce food, while others reproduce through specialized partnerships that would seem improbable in shallow water. Communities gather around whale falls, hydrothermal vents, and other isolated sources of energy, creating ecological networks that can persist without any direct dependence on sunlight reaching the seafloor. The result is an ocean within the ocean, governed by its own constraints and opportunities. Food arrives from above. Chemicals rise from geological activity. Animals detect one another through pressure, electricity, scent, and bioluminescence. And bodies are shaped by forces that remain almost absent from our everyday experience. Nothing about this system requires the surface to serve as its model. For hundreds of millions of years, these environments have provided the setting in which their inhabitants evolved, reproduced, competed, and disappeared, leaving descendants behind. Their bodies carry that history in every living tissue. What appears strange from above is therefore not strange to the lineage itself. The transparent tissues, reduced bones, enormous eyes, missing eyes, flexible bodies, and unusual senses are pieces of a biological language written by pressure, chemistry, scarcity, and darkness. The deep sea was never a place these creatures had to become accustomed to. It was the place that made them what they are. An evolutionary arms race leaves no final design. Every advantage changes the conditions faced by everything around it, and every response becomes part of the environment in which the next response evolves. In the deep ocean, that process has been unfolding across immense stretches of time, leaving behind bodies that seem improbable only when viewed without their history. Light is one of the clearest examples. A predator produces a glow that helps it find food, and prey that survives encounters with that glow becomes part of the next generation. Over time, sensitivity itself becomes a battleground. Some animals learn to detect faint differences in brightness, while others reshape their bodies so their own illumination becomes harder to distinguish from the water around them. Counter-illumination turns the glow of an animal into camouflage, while specialized eyes evolve to separate those faint signals from the surrounding darkness. The strawberry squid carries this contest in its mismatched eyes. Its enormous upward-facing eye gathers the sparse light produced above, while the smaller yellow eye points downward toward the faint bioluminescence of potential prey. Neither eye is simply better. Each is tuned to a different direction in the same optical landscape. The barreleye follows another path, with tubular eyes and a transparent shield that allows its vision to survey the water above while protecting delicate tissues. These systems are not isolated inventions. They are responses shaped by generations of animals detecting one another more effectively. Then there is the red light of the dragonfish. Most deep ocean animals cannot see this wavelength, allowing the dragonfish to illuminate its surroundings without announcing itself in the usual way. Yet, the advantage exists only while its prey remains unable to detect it. Any mutation that gives a prey animal even a small improvement in sensitivity could begin shifting that balance. A useful adaptation, therefore, carries the seed of its own limitation. The goblin shark reveals the same principle through movement. It does not need the streamlined body or rapid pursuit associated with open water hunters. Instead, its extraordinary jaws can extend toward prey with remarkable speed, converting a slow-moving predator into something capable of a sudden capture. The jaw is not an isolated curiosity. It is a solution to a particular ecological problem, and its existence reflects the pressures created by the behavior of other animals. Among anglerfish, scarcity changes the contest again. Finding a mate in an enormous habitat can be so unlikely that the male has evolved toward permanent attachment to the female in some species. Independent existence becomes less valuable than certainty of reproduction. What appears extreme from the outside is therefore another response to the conditions imposed by population density, distance, and the difficulty of encountering another member of the same species. None of these adaptations exists in isolation. A new sensory ability changes predation. Changed predation alters prey behavior. New prey behavior changes the value of camouflage. Better camouflage favors sharper detection. The cycle continues, not toward perfection, but toward temporary solutions that remain useful until the surrounding biology changes again. Hundreds of millions of years can pass within this exchange, yet there is no final winner waiting at the end. Evolution does not produce a finished creature. It produces organisms that fit, for a time, into a moving network of pressures and opportunities. The enormous eye, the retractable jaw, the strange red glow, the permanent mating bond, and every other unusual feature are traces of negotiations that never truly ended. Seen this way, a bizarre animal is not a failed attempt at being something familiar. It is a record of what succeeded under conditions that demanded something different. The mistake was never in the animal. The mistake was in assuming that every creature should resemble the world we already understand. A mistake can exist only when there is something called correct. The word carries an invisible blueprint, a shape imagined before the living thing appears. Yet nothing in evolution begins with a blueprint. There is no master form waiting beneath the surface, no ideal body toward which every lineage is moving. There are only organisms, environments, inheritance, variation, and the long filtering process of survival and reproduction. That distinction changes the meaning of almost every creature we have encountered. The blobfish was judged against the proportions of animals that evolved in air and shallow water, where muscles support bodies and gravity pulls differently through the surrounding medium. Its soft tissues make sense within the pressures of the deep ocean. What looked malformed when removed from that setting was never designed for ours. The apparent defect belonged to the observer, not the animal. Our sense of beauty is also biological history. Human faces are familiar because human brains developed among faces. Eyes usually appear near the front of a head. Mouths occupy a recognizable position, and bodies tend to preserve proportions that our visual systems learn to recognize across countless generations. Those preferences are useful in our own world. They are not laws of nature. Far below the surface, evolution has had other materials to work with. Darkness changes the value of vision. Scarce food changes the value of energy. Immense pressure changes the architecture of tissues. Vast distances between potential mates change reproduction. The resulting forms can seem strange because they solve problems that our bodies never evolved to encounter. The anglerfish offers perhaps the clearest example. In some species, the male becomes permanently attached to the female, eventually sharing circulation with her. From the perspective of ordinary human anatomy, the arrangement appears almost impossible, yet the behavior arose within a particular reproductive landscape, where finding another mate can be extraordinarily difficult. The form is not a failed version of courtship, it is a solution produced by a very different set of pressures. Even the word grotesque reveals a boundary in human perception. It describes something that departs from an expected shape, which means the observer must already possess an expectation. A transparent body, a missing structure, an enormous eye, or a jaw that extends beyond the head becomes disturbing only when measured against a familiar template. Evolution does not measure organisms against templates. It has no preference for symmetry, elegance, recognizability, or resemblance to us. A feature persists when it contributes enough to survival or reproduction to remain in the lineage. Sometimes that contribution is direct. Sometimes it belongs to a complicated chain of relationships involving predators, prey, mates, microbes, temperature, chemistry, and the availability of food. This is why the deep ocean can produce animals that look unfinished while being exquisitely fitted to their surroundings. Their forms are records of pressures that accumulated across immense spans of time. Each strange contour carries information about what the environment demanded and what earlier generations could preserve. The deeper lesson is not that beauty is meaningless, nor that every unusual organism must be admired. It is that appearance is evidence, not judgment. A body can seem wrong and still be extraordinarily successful. A form can look fragile while representing millions of years of persistence. What we call ugly may simply be a biological language written for a world our senses were never built to inhabit. The mistake, then, lies in confusing familiarity with correctness. We inherited a narrow visual vocabulary from life on the surface, and the deep ocean keeps showing us that nature was never required to speak it at all. Look closely at the bodies gathered across this story, and a different pattern begins to emerge. The strange shapes are not collections of defects. They are records of problems that had to be solved, each written into flesh over generations, with the surrounding ocean shaping the answer. The blobfish carries perhaps the simplest version. Its tissues contain large amounts of water, with reduced muscle and a body structure suited to tremendous pressure rather than supporting itself in air. At great depth, buoyancy helps support its soft form, reducing the need for dense bones and powerful muscles. What appears collapsed when brought upward is therefore a poor representation of the animal in its natural setting. Its softness is not an absence of design, it is the design. The barreleye takes another path. Beneath its transparent shield, its tubular eyes can rotate upward toward faint light and forward when feeding. The dome protects delicate tissues while allowing the animal to gather information from above. In a body only about 15 centimeters, about 6 inches long, several functions occupy the same compact space. Protection, light collection, and visual targeting become parts of one structure. For the goblin shark, the problem is distance. Its body is not built for rapid pursuit, yet prey can pass beyond the reach of an ordinary bite. The jaws, therefore, travel outward with remarkable speed, extending far beyond the front of the head before closing around prey. Reach replaces pursuit. A movement that looks theatrical becomes a precise mechanical solution when speed is expensive and opportunity is fleeting. The gulper eel approaches scarcity differently. Its mouth can expand into a cavernous opening, creating an enormous capture area compared with its slender body. A glowing structure at the end of its tail adds another possibility, producing light in a habitat where a small luminous point can become a useful signal. The animal does not need to search constantly. Its anatomy allows chance encounters to become meals. Among anglerfish, efficiency becomes even more concentrated. The female carries a lure powered by bioluminescent bacteria, bringing prey within reach while minimizing pursuit. Inward-facing teeth help prevent captured animals from escaping. The system combines attraction, timing, and restraint into one hunting apparatus. The male solves an entirely different problem. In some species, finding a mate in the immense deep ocean may be so unlikely that temporary contact is an evolutionary gamble. Permanent attachment removes that uncertainty. His body changes after joining the female, and in some species becomes largely devoted to reproduction. What seems like disappearance of individuality is instead a reproductive strategy shaped by extraordinary scarcity. Elsewhere, the giant isopod adjusts to a world where meals may arrive after long intervals. Its metabolism can remain low, allowing it to endure extended fasting and use large food falls when they occur. The hatchetfish answers another threat, using light from its underside to blend with faint illumination above, reducing the outline visible to predators below. The dragonfish takes the opposite approach. Its red bioluminescence reaches wavelengths that many nearby deep-sea animals cannot detect, giving it a private beam in a world where most creatures are sensitive mainly to shorter wavelengths. One animal hides with light, another hunts with it, both exploit the same environment from different directions. Across these bodies, evolution has not been drawing toward beauty, strength, or familiarity. It has been solving local problems with whatever materials inheritance made available. Softness answers pressure. Reach answers slow movement. Enlargement answers scarcity. Attachment answers distance. Hidden light answers hidden vision. Every feature is an answer, and the answer only makes sense when the environment is included. Remove the environment, and the body can look like a mistake. Put the body back into the world that shaped it, and those strange proportions begin to read as biological grammar, each unusual structure carrying the history of a problem solved long before we arrived to name it. For hundreds of millions of years, the deep ocean has been conducting an experiment without a laboratory, a blueprint, or an observer waiting beside it. The conditions were never designed to produce elegance. They simply persisted, and life kept meeting them with new forms. A dragonfish eventually acquired the ability to produce red light, opening a private channel of illumination in a world where many potential prey cannot perceive it. An anglerfish lineage altered its immune system so profoundly that permanent tissue fusion became possible. A barreleye developed eyes that could rotate within a transparent shield, allowing its vision to serve more than one purpose. Siphonophores took cooperation so far that a colony could function as something larger than any individual body. In the deepest trenches, snailfish accumulated molecules that helped their proteins remain functional beneath immense pressure. None of these solutions appeared because an organism understood the problem. There was no intention behind them, only variation followed by selection. A slight advantage could persist. A disadvantage could disappear with the lineage that carried it. Generation after generation, the environment became a filter through which countless possibilities passed. That process becomes difficult to imagine because the timescale is so much larger than a human life. Complex animal life has been exploring the deep ocean for roughly 500 million years, beginning around the Cambrian period, when animal diversity expanded dramatically. Across that immense interval, lineages encountered changing chemistry, shifting food supplies, predators, competitors, temperature, pressure, and the absence of sunlight. Each generation inherited a little of what came before, then added its own small variation. The goblin shark lineage reaches back more than 120 million years, long before the first familiar mammals dominated the land. The body plan of giant isopods belongs to an ancient branch whose relatives were already established around the time dinosaurs began appearing. The vampire squid represents another persistence across deep evolutionary time, surviving while related branches vanished and ecosystems transformed around it. What remains is not a record of perfection. Evolution leaves behind compromises, abandoned structures, peculiar proportions, and solutions that work only within very specific circumstances. Yet the accumulated result can resemble invention, because the fit between organism and environment becomes so intricate. A structure is shaped by one pressure, then another, until the final form carries traces of an entire history within its tissues. The scale becomes even stranger when we consider how little of that history has been witnessed directly. In the Clarion-Clipperton zone, surveys have repeatedly revealed animals unknown to science, with one major study finding that more than 90% of collected specimens represented species not previously described. The number is remarkable, but the deeper meaning lies in what it implies about the unseen majority. We are not standing at the end of a catalog. We are listening near its first pages. For most of human history, the deep ocean was present only as an unseen volume beneath the waves. Now, cameras, submersibles, genetic analysis, and pressure resistant instruments are beginning to reveal fragments of a biological history that never depended upon our presence. 500 million years of selection have been unfolding without an audience. The forms we see today are not the beginning of that story, and they are certainly not its final expression. Each one is a brief frame in a process whose older chapters remain beyond our reach. They are temporary arrangements in a process still moving, carrying solutions older than our species through a world that has been alive for longer than our imagination can comfortably hold. For now, beneath the waves, below the reach of daylight, life is continuing without any awareness of the shore above it. The deep ocean does not pause for our attention, and its living rhythms continue beneath us whether anyone is watching or not. Currents move through trenches and plains, particles descend from distant waters, and organisms continue the routines that shaped their bodies long before human beings could imagine such places. Somewhere more than 3,000 feet, about 914 meters, beneath the surface, a barreleye may be holding its position with delicate adjustments of its fins, its tubular eyes angled toward the faint traces above. Elsewhere, a goblin shark may be moving through the water with its long snout, sensing disturbances and electrical signals produced by nearby prey. Neither scene requires an audience. Each belongs to a world that existed before observation became possible. A giant isopod may remain for long periods with very little food, its metabolism matched to the irregular arrival of nourishment from above. When a meal finally reaches the sea floor, an entire community can gather around it, transforming one fallen body into energy that passes through many forms of life. The event is not unusual enough to be miraculous. It is part of the ordinary rhythm of an ecosystem built around scarcity. Farther into the oxygen-poor waters, a vampire squid may extend its delicate feeding filaments through drifting particles, collecting fragments that once belonged to organisms living much closer to the surface. A hatchetfish may use light produced along its underside to reduce the outline visible from below, turning illumination into a form of concealment. Nearby, an anglerfish may carry its living lure through the darkness, allowing bacteria to provide the light while the fish conserves the energy that pursuit would require. None of these animals evolved with the possibility of being studied. For nearly the entire history of their lineages, there were no cameras, submersibles, research vessels, or human eyes capable of reaching them. Their ancestors lived and died without knowing that another world existed above the water column, just as the generations that followed had no reason to anticipate the arrival of instruments descending from the surface. That separation gives the deep ocean a particular kind of scale. It is not simply a place far away. It is a vast biological realm whose history unfolded independently of human attention. The animals living there do not perform their strange behaviors for us, and their bodies were not shaped with our ideas of beauty, efficiency, or familiarity in mind. The ocean covers about 71% of Earth's surface, yet enormous portions remain difficult to observe directly. New expeditions continue to find species, behaviors, and biological relationships that were absent from earlier records. Each discovery adds another small piece to a living system that was already complete before we arrived to name its inhabitants. And perhaps that is what these creatures leave with us, not a lesson that belongs only to the deep, but a change in proportion. The familiar world around us is real, but it is not the whole world. Beyond the limits of ordinary experience, other forms of life continue their ancient routines, sustained by conditions that would be impossible for us to endure. They do not need our understanding to continue. They do not need our presence to make their existence meaningful. Their lives were already underway, extending through generations, long before the first human imagination reached down into the darkness. And while we sleep beneath the surface of our own small lives, that older world continues below, carrying on without waiting for us. The title began with a judgment hidden inside a simple word: mistake. It suggested that somewhere along the long history of life, evolution had taken a wrong turn and produced creatures that seemed unfinished, excessive, or strangely assembled. Yet after following their bodies into the conditions that shaped them, that judgment becomes difficult to hold. To call something wrong, we first need an image of what right should look like. Human beings carry that image with them everywhere. We recognize familiar proportions, familiar faces, familiar ways of moving through air and sunlight. Our senses were formed on the surface, where vision travels easily, where gravity is gentle, and where food can be abundant enough to support bodies built for pursuit and speed. Far below, those expectations lose their usefulness. At 200 meters, about 656 feet, sunlight has already begun to fade into a different biological world. Deeper still, pressure rises with every descent, and the conditions that shaped surface animals give way to another set of demands. The body is no longer being judged by appearance, it is being tested by physics. The barreleye offers one of the clearest examples. Its transparent head allows its tubular eyes to gather the faint illumination arriving from above, while the eyes can rotate when the animal shifts from searching to feeding. What looks extraordinary from our perspective becomes an integrated arrangement when viewed through the conditions of its habitat. The shape is not an accident waiting to be corrected, it is a structure that works. The same principle reaches across the deep ocean. A goblin shark does not need a streamlined body designed for long pursuit when its feeding strategy allows the jaws to travel outward toward prey. An anglerfish does not need to search constantly for a mate when its lineage has developed a reproductive system suited to encounters that may be extraordinarily rare. A snailfish does not require the rigid architecture familiar from many shallow water fishes when its tissues and chemistry are suited to intense pressure. None of these forms was planned in advance. Evolution does not imagine a finished creature and then construct it. Small inherited differences appear. Environments filter those differences, and successful traits become more common across generations. Given enough time, that process can produce structures that seem almost deliberate, even though no designer ever stood behind them. The deep ocean, therefore, gives us something more valuable than a collection of strange animals. It gives us another frame for understanding life itself. Biology is not a contest to approach one ideal body. It is a history of relationships between organisms and the conditions around them. Change the conditions, and different solutions become possible. That is why the word mistake belongs less to these animals than to the assumptions we carry down with us. We looked at unfamiliar bodies and measured them against a standard created by our own surroundings. Their forms appeared incomplete because our reference point was incomplete. For hundreds of millions of years, the deep ocean has been shaping life without asking whether its creatures resemble anything above. It has produced bodies that float, glow, stretch, sense, merge, wait, and endure, according to pressures and opportunities particular to that realm. So the deepest lesson is not that evolution never makes mistakes. It is that nature does not owe us a familiar definition of correctness. A body can look impossible and still be perfectly fitted to its world. A creature can seem strange because its history is written in conditions we rarely experience. The animals we called mistakes were never unfinished versions of something better. They were finished answers to problems we had not yet learned to see.